I want to go ahead and show you the tools that we're going to use today. The first tool that we're going to use for measuring pH is a pH meter. So to do our pH lab, we're going to be using this pH meter today. And the first thing that we'll want to do is to calibrate it with solutions of known pH. So today we're going to use a solution of known pH 4 and known pH 7. So I'm going to go ahead and show you the glass bulb, which is what is actually doing the measuring of the solution. And you always, between uses, you always want to rinse with deionized water and then wipe it with a Kim wipe. And a Kim wipe is something you'll find in every lab. It's kind of like a lint-free tissue. So I'm going to go ahead and turn the pH meter on. I'm going to put it into calibration mode. There are a list of directions for every pH meter that you use. This happens to be the directions for the one that we're using today. But calibrating a pH meter is pretty similar regardless of the model of pH meter you use. So as you can see, it's been sitting in the pH 4 solution for a couple of minutes, and right now it's reading 4.18. The lower scale says 4.01, and when I push the calibrate button, it will switch to that 4.01. And then I'm going to rinse, dab it with the Kim wipe, and now I'm going to put it in the solution of known 7.0 solution. So right now it's reading 6.92. I'll give it a little bit to equilibrate till where that number stops switching, and then we will calibrate it to 7.0. Okay, so right now it reads 6.99, so it's already pretty calibrated, but I will complete the calibration and rinse the pH meter. Okay, I think we're done. Okay, so now that we're done with the pH standard solutions, I'm going to go ahead and empty those into the liquid waste container. We never dump anything that isn't water down the sink in a science class. So we will give these a little rinse. And the next step is to actually begin the experiment. So the next step of the lab is to measure out our water and take the pH of that. So first thing I'll do is I will measure 20 milliliters of water using this graduated cylinder. And you have likely learned that when you measure a liquid in a graduated cylinder, you want to look at the meniscus. So we can take a look at that. Here's a close-up of what that meniscus looks like. You never want to use the measures on the side of a beaker for anything besides just an estimate. You need a graduated cylinder to be more accurate. So I'm going to go ahead and pour that 20 mils of water in my beaker, and then we're going to take the pH of that. So I want to show you this fun piece of lab equipment. It is a magnetic stirrer. So I'm going to put the beaker on the magnetic stirrer, and then I need a stir bar. So here's the stir bar. It's a little magnet, and this is a stir bar retriever. So we'll take our stir bar and place it gently in the beaker, and then put the beaker back on the stir plate and then let it stir. So you'll see that as we turn it on, it begins to stir. That's a little fast, so we don't need it to go that fast. And we don't really need that yet, but I just wanted to show it to you. I'm going to go ahead and turn it off so I can take the pH of the solution. So the pH meter turned off, but that's okay. It's still calibrated. So when I take the pH of this, well, before I tell you what the meter says, what do you expect the pH of the water to be? Your lab may ask you this, so go ahead and write it down if it asks. And I don't know that you can see that on video, but it says 6.29. That might be different than what you were expecting. So does anybody have any ideas about why that might be? Why that pH might be lower than the 7.0 that you might have been expecting for water? So if you're not sure of the answer to that, I will tell you that carbon dioxide from the atmosphere does dissolve into water, and depending on what the pH of it is to begin with, it can reduce the pH. So let's go ahead and add solution A to our water and see how it affects the pH. So to do that, I'm going to use this pipette. And this happens to be a 5 ml pipette. You can see the measures here, 0, 1, 2, 3, 4, and then 5. This is a pipette pump, and I'm going to put the pipette pump onto the pipette. I want you to think about how you would expect the pH of this water to change if we add an acid, or if we add a base, or if we add a salt. So let's go ahead and give it a try. What I've done is I've poured just 10 to 15 milliliters of A, B, and C in these beakers, just an approximation. Now we're going to add our unknown solution A. So I'm going to use a pipette and pipette pump to measure out the 5 mils of solution A. So I'll put the pipette pump on the pipette, and then I'm just going to roll up, which you can see draws up the solution A until I have measured 5 mils, which the line is right here, and then I'm going to simply do the reverse to add the solution to the beaker. And I have it stirring, not particularly fast, and now we are ready to measure the pH of that solution. So I'll go ahead and turn this off, put the pH meter in, and we can see what the pH of our solution, how it changes after we've added solution A. And the number is 3.89. So that is a decrease in pH. So what does that tell us about whether solution A was an acid, a base, or a salt? Okay, so I'm going to go ahead now and rinse the pH meter. And then I'm going to add 5 mils of solution B. Now I could be using a graduated cylinder, a small graduated cylinder, to measure out the 5 mils, but the pipettes are a little more accurate at these smaller volumes. Okay, so we'll get it stirring. Add solution B. And we don't know whether solution B is an acid, a base, or a salt, but we will be able to determine that by taking the pH. So I'm going to go ahead and put the pH meter in here before. So remember, our pH before was low, 3.92, I believe. And right now, it is reading 4.18. So I will then rinse our pH meter again, and then we'll add solution C. And you'll notice that I'm not emptying the beaker between adding solution A, B, and C. We're really just looking for a relative change in pH. So before, it was a significant drop. And you can see those numbers here on the slide. For solution B, you can see on the slide how it changed a little bit, but not as dramatically. So now let's see what solution C is going to do to the solution. Okay, so now go ahead, Now we will go ahead and measure solution C. 5 mils of that. And add it to our solution, and we will see how that affects the pH. Okay, I don't know if you can see that or not, but it looks like it's still changing, but right now it's at 6.45, and it's continuing to go up. And it looks like it's leveling off at 6.63. So we are done with that first part of the experiment. And from the data that we've generated, you should be able to determine which one is an acid, a solution that is going to add hydrogen ions to a solution and reduce the pH, which one of these three, A, B, or C, is a base, is going to remove hydrogen ions and is going to increase the pH, and which one is a salt, which is going to not significantly affect the pH of the solution. So now that we're done with that, we can go ahead and remove our magnetic stirrer with our stir bar remover, and then empty this into the liquid waste. Okay. So now that we know what solutions A, B, and C are, which one's an acid, which one's a base, and which one's a salt, now we're going to see how those an acid, a base, and a salt react in buffer. So we know that living systems are buffered, and as you likely know, a buffer is something that resists change in pH. So your lab likely has a place for you to write down some predictions of how you expect solution A to behave in a buffer versus in the water, how you expect solution B and solution C to behave in the buffer. So go ahead and make those predictions if you haven't already. What I'm going to do now is I'll measure out the 20 mils of the buffer, just like we measured out the 20 mils of water before. And I'll add this to our beaker. And we'll put our stir bar back in. I don't think you saw me rinse it, but I did rinse it since it had been in our previous solution. And now we're going to take the pH of the plain buffer and give it a little bit to stabilize. And its pH is higher than water. It looks like it's stabilizing at, just jumped up a little bit. Usually you want to let it be stable for 3 or 4 seconds before you call that number. And it is still continuing to move, but it looks like it's stable at 8.03. So that's what we're starting with. So when we add solution A, how do you expect it to change? Remember, solution A before decreased the pH of the water. So do you think it's going to increase or decrease the pH of this buffer? And if so, do you think it's going to increase it or decrease it an equal amount or less? So the pH for this is 7.82. And now we're going to add solution B. I want you to be thinking about how you expect that to affect the pH of this solution. All right, and it looks like it has stabilized at 7.97. And last but not least, we will measure solution C. And you've noticed that I'm changing the pipettes and using a clean one each time, and that's of course good practice. And for our last measurement, what do you expect to have the pH, how do you expect it to change, to increase, decrease, or stay about the same? And I will tell you that it has changed to 8.25. All right, so we'll go ahead and clean up. You don't have to watch that part, but I just want to refer you to your lab handout where you're answering questions, and you're going to be asked if your results supported your predictions or not. So just to review the goals of this lab today, the objectives are here, but we want to make sure everybody's got a firm grounding in understanding pH, because when we start to learn about biological molecules like carbohydrates and fats and proteins, we will see that pH can affect them. I want you to think about your refrigerator, and I want you to think if you have any ketchup in there, or maybe a jar of pickles. And if you have those items in your fridge, have they been there a while? And if they have, it's okay. They can last months or even years sometimes, because those foods are highly acidic. And the acid, the low pH, helps to denature or change the proteins that food spoiling organisms like bacteria use. So if you have bacteria that lands in your food, but their enzymes to break down the food get denatured, that means the food gets preserved. So foods that are highly acidic can last a long time. So I thank you for your time and attention, and hope you have a good rest of the day.